Okay, so in this experiment B, we're going to investigate a little bit about a anaerobic form of cellular respiration that we've learned about called fermentation. And in this experiment, we're going to use some yeast cells and investigate whether they can indeed do some fermentation, and also give them different choices of energy-rich organic molecules to potentially try to use to do their fermentation with. In our textbook examples, there were using glucose as the example molecule, but maybe they can use other molecules as well. So we have a setup here of different solutions with different organic molecules for the yeast to potentially try to use for fermentation. And what we're going to do is take a little bit of yeast, about 10 milliliters, measured in the graduated cylinder, and we're going to use what are called fermentation tubes, which makes sense if we're going to be looking for fermentation, to do the experiment with. We're going to load each one of these tubes with the portion of yeast, like so. Then we're going to give them a potential energy-rich molecule to work with, in this case, glucose here. And again, we're going to use another 10 milliliters of this solution added to the yeast to give them something to potentially work with. Once you've added the proper amount of solution, we need to tip the little fermentation tube so that this end, that is a blind end to the tube, doesn't have any air left in it. So a little bit of manipulation like that, and you see it's all fluid. And we're going to use beakers to hold these tubes because they won't stand up on their own on a tabletop. And we're also going to add a little bit of warm water, kind of like a little warm bath for the setup here, and then we'll give them a chance to go to work. Okay, so once you have your fermentation tubes all loaded and placed in the beakers with a little bit of a water bath, we're going to allow the yeast a portion of time to go to work on that molecule that you've given them to try to use for fermentation. And to help speed things along a little bit, we're going to place our beakers that are ready to go on a warming tray like you see here, so that they can be nice and warm and get to business with their fermentation. And the way we're going to measure whether they're doing any fermentation or not is looking for one of the waste products that we've learned about that happens with fermentation, CO2 gas being given off. With that blind end of the fermentation tube, the CO2 bubbles that do get produced, if they get produced, will collect at that end, and we can do a quick measure of how much collects during the time period that we're working with at every increment indicated in the lab manual directions, and see which produces the most gas bubbles, which we would assume is giving us an indication of the most fermentation being done.